The motor unit is discussed in this screencast. This topic may be found in Chapter 6 of your textbook. This screencast was designed to achieve the following objectives. Describe a motor unit. Explain how motor units differ in muscles with fine motor control versus in muscles with coarse motor control. Describe in detail a neuromuscular junction. Describe in detail how an action potential produced in a motor neuron causes contraction in a skeletal muscle fiber. And, lastly, describe how a skeletal muscle fiber relaxes. In the previous screencast, you learned that contraction of a skeletal muscle is based on the contraction of shortening of a sarcomere. So here we have a sarcomere at rest when the myosin heads of the thick filaments interact with the thin filaments and pull them inward. It causes the thin filaments to slide inward and you have contraction of the sarcomere. Contraction of the sarcomere leads to contraction of the entire myofibril. Contraction of all the myofibrils of the skeletal muscle fiber causes the skeletal muscle fiber as a whole to contract. And as all the skeletal muscle fibers of a muscle contract, that muscle, the organ, contracts. So now that you know that the basis of skeletal muscle contraction is the interaction between the myosin heads and the thin filaments of the sarcomeres, the obvious question is, well, what causes the myosin heads to bind to the thin filaments in the first place? And the answer is stimulation by a motor neuron initiates the interaction between the myosin heads and the thin filaments. Recall that muscle fibers have a property called excitability. Motor neurons, through the release of neurotransmitters, cause changes in the skeletal muscle fiber, which causes the myosin heads to interact with the thin filaments. A motor unit is a motor neuron and all of the skeletal muscle fibers that it stimulates. Here we have a skeletal muscle composed of 3 motor units. You have 3 motor neurons in the anterior horn of gray matter, each forming junctions with several skeletal muscle fibers of the skeletal muscle. The motor neuron of motor unit A forms junctions with the skeletal muscle fibers that are color-coded purple. The motor neuron of motor unit B forms junctions with the skeletal muscle fibers color-coded in blue. And the motor neuron of motor unit C forms junctions with the skeletal muscle fibers color-coded in green. When an action potential moves down the axon of a motor neuron, it will cause contraction of all the skeletal muscle fibers of the motor unit. So the entire motor unit contracts and relaxes at the same time. Motor units can be composed of just a few skeletal muscle fibers or large numbers of skeletal muscle fibers. Muscles that require small, highly precise movements, such as the muscles that control eye movement, may have as few as 3 muscle fibers per motor unit. However, muscles responsible for large, powerful movements, such as the muscles of the thigh, may have several thousand muscle fibers per motor unit. In certain parts of the body, precise control of movement is much more important than, say, power. We say that these parts of the body exhibit fine motor control. Some examples would be the muscles of facial expression and the muscles that control the hands, the fingers, the position and movement of the wrist. In these areas, since power is not important and precise control of movement is important, there tends to be lots of small skeletal muscles. Therefore, consequently, the motor units tend to be small as well, perhaps only 2 or 3, 4 to 6 skeletal muscle fibers per motor neuron. So in areas of the body that exhibit fine motor control, motor units tend to be rather small. Contrast that with parts of the body where precise control of skeletal muscle movement is less important and power is much more important, such as the lower appendages. In your lower appendages, your thigh and your leg, you have relatively large skeletal muscles. There are fewer of them, and consequently, the motor units tend to be rather large. You can have one motor neuron controlling several hundred skeletal muscle fibers. So in areas of the body where you have coarse motor control, motor units tend to be rather large. Where axons interact with skeletal muscle fibers, we have what are called neuromuscular junctions. So if we were to zero in on a neuromuscular junction and we looked at it under the microscope, we'd see an image that looks like this. Here you can see the various axons and you can see where they form neuromuscular junctions with various skeletal muscle fibers. Now if we look more in depth at a neuromuscular junction, we observe that the axon actually divides into several axon terminals at a neuromuscular junction. Also notice that there is an enlarged end of the axon terminals called a synaptic bulb. Before discussing what occurs at a neuromuscular junction, I want to look at the anatomy of the neuromuscular junction. So here we have a myelinated axon of the motor neuron. That axon is enlarged at the end into a synaptic bulb. I want you to notice that the axon and the sarcolemma of the skeletal muscle fiber do not actually come into contact with one another. There's a little space there filled with extracellular fluid called the synaptic cleft. I also want to bring to your attention the many vesicles in the synaptic bulb. These vesicles contain the neurotransmitter acetylcholine. Remember that the somatic nervous system releases exclusively the neurotransmitter acetylcholine. Now let's discuss the events that occur at a neuromuscular junction. So let's say that a motor neuron is going to stimulate a skeletal muscle fiber to contract. Let's look at the events that are involved in that process. So the motor neuron is sending an action potential or nerve impulse down its axon. It's going to move into the synaptic bulb. When it does so, it causes the vesicles of acetylcholine to merge with the plasma membrane and dump their contents, the acetylcholine, into the synaptic cleft. Now, some of that acetylcholine is going to be degraded by an enzyme called acetylcholinesterase, but some of that acetylcholine will bind receptors on the plasma membrane of the skeletal muscle fibers. When that acetylcholine binds to those receptors, it causes sodium channels to open. As those sodium channels open, sodium moves into the cell and causes a depolarization that leads to the production of an action potential. Basically, by the same mechanism that we saw action potentials produced in a neuron. As that action potential spreads throughout the sarcolemma of the skeletal muscle and down the T-tubules, it causes a release of calcium from the sarcoplasmic reticulum. So you may ask the question, what in the world does the release of calcium from the sarcoplasmic reticulum have to do with muscle contraction? Well, I'm going to explain that now. So here we have a sarcomere. Here's our thick filament with the myosin heads shown in red, and here are thin filaments shown in blue. There are these protein complexes on the thin filaments. Your book gives them a name, troponin, but I am not requiring you to know the name. Just know that they're protein complexes, and know that normally they prevent the myosin heads from accessing the myosin binding sites on the thin filaments. So these proteins block the ability of the myosin heads to bind the thin filaments. And so if the myosin heads can't bind the thin filaments, they can't contract the sarcomere. So when an action potential causes calcium to be released from the sarcoplasmic reticulum, that calcium binds to these protein complexes, moving them out of the way, opening up the myosin binding sites. The myosin heads are then able to access the binding sites on the thin filaments. After the myosin heads bind to the thin filaments, they pivot, moving the filaments toward the center of the sarcomere. ATP then allows for those myosin heads to detach and reposition for binding again. And as long as calcium is present, they will bind again, pivot, moving those filaments toward the center of the sarcomere, and ATP, as long as it's present, will allow them to detach again. So you have this repeated cycle of binding, pivot, detach, reposition, binding, pivot, detach, reposition, which moves those thin filaments toward the middle of the sarcomere, causing contraction of the sarcomere, which leads to contraction of the skeletal muscle cell as a whole. Notice that both calcium and ATP are required for skeletal muscle contraction. The calcium is responsible for opening up the binding sites, allowing the myosin heads to attach to the thin filaments. However, ATP is required for the myosin heads to detach and reposition themselves for reattachment. This figure from your book illustrates what your book calls the cross-bridge cycle. A cross-bridge is nothing more than what is formed when the myosin head binds the thin filament. That is referred to as a cross-bridge. So here we have a sarcomere at rest. Notice that the myosin head is not binding the myosin binding site on the thin filament. This is because there's no calcium available, and therefore the protein complex known as troponin is obscuring the access of the myosin head to its binding site on the thin filament. Now let's say that an action potential that is caused by the release of a neurotransmitter from a motor neuron causes the sarcoplasmic reticulum to release calcium. So calcium is released into the cytoplasm. That calcium binds to the troponin, and it moves that complex out of the way, opening up the binding sites for the myosin. So myosin forms the cross bridge, binding to its binding site on the thin filaments. The myosin heads then pivot and move the thin filaments toward the center of the sarcomere. This process is called the power stroke. And if ATP is present, the ATP will cause the myosin heads to detach and reposition. And as long as calcium is present, the myosin heads will reform the cross bridges, cause the power stroke again, detach in the presence of ATP, reposition for another formation of a cross bridge. And so the cycle of attach, pivot, release, attach, pivot, release, which requires calcium for attachment and ATP for release, causes the contraction of the sarcomere, which leads to contraction of the skeletal muscle fiber as a whole. Now I want you to note that when calcium is released from the sarcoplasmic reticulum, it is immediately taken back up by the sarcoplasmic reticulum. So it takes continuous stimulation by a motor neuron for a continuous release of calcium for this cycle to continue. Whenever a motor neuron ceases to release neurotransmitter, the calcium is taken back up by the sarcoplasmic reticulum, and now the myosin heads are no longer able to bind to their binding sites on the thin filaments. And that is what leads to the relaxation of a muscle. So let's review how a motor neuron stimulates a skeletal muscle fiber to contract. An action potential moves down the axon, enters the synaptic bulb. There, it causes vesicles containing acetylcholine to dump the acetylcholine into the synaptic cleft by exocytosis. Some of the acetylcholine is degraded by acetylcholinesterase. Some of the acetylcholine binds to receptors on the sarcolemma of the skeletal muscle fiber. The binding of acetylcholine to the receptors causes sodium channels to open. As sodium channels open, sodium moves into the cytoplasm of the cell, causing an action potential, and that action potential causes the release of calcium. With calcium now available, calcium binds to the protein complexes, causing the myosin binding sites to now become available for the myosin heads to form cross bridges. As the myosin heads form cross bridges, they pivot, producing the power stroke as the thin filaments are pulled inward toward the center of the sarcomere. That causes the sarcomere to shorten. If ATP is present, the myosin heads can then detach and reposition for another round of cross bridge formation. As long as the motor neuron continues to release neurotransmitter, there will be a continuous production of action potentials in the skeletal muscle fiber and a continual release of calcium to allow for muscle contraction. As soon as the motor neuron ceases to release neurotransmitter, most of the calcium will move back into the sarcoplasmic reticulum, and now the troponin moves back to its original position, blocking the myosin binding site, and therefore muscle relaxation occurs. This is a figure from your book illustrating muscle contraction on the left and muscle relaxation on the right. Admittedly, this is a bit redundant and repeats the same information from the two previous slides. So if you have a good understanding of muscle contraction and relaxation as affected by the release of neurotransmitters from a motor neuron, you might want to skip the slide. So on the left, here we have an action potential traveling down an axon of a motor neuron. When that action potential enters the synaptic bulb, it causes vesicles containing acetylcholine to dump the acetylcholine into the synaptic cleft. The acetylcholine diffuses across the synaptic cleft and binds to receptors on the plasma membrane of the skeletal muscle fiber. This causes sodium channels to open and an action potential is initiated. That action potential moves across the entire sarcolemma down into the T-tubules and causes the sarcoplasmic reticulum to release calcium. Calcium binds to protein complexes on the thin filament, revealing the myosin binding sites, so the myosin heads then bind their binding sites on the thin filaments, forming cross bridges. When they form cross bridges, they pivot, causing the power stroke. If ATP is available, they release and reposition, and as long as calcium is still being released by the sarcoplasmic reticulum, they can form additional cross bridges and muscle contraction continues. Muscle relaxation follows a ceasing of nerve impulses moving down the axon of a motor neuron. So if action potentials no longer move down the axon of a motor neuron, there is no longer the release of acetylcholine from the synaptic bulb of the motor neuron. Without the release of additional acetylcholine, the sodium channels close, and we no longer have action potentials being produced. In the absence of action potentials, all of the calcium, or most of the calcium, in the cytoplasm is transported back into the sarcoplasmic reticulum. In the absence of calcium in the sarcoplasmic reticulum, the binding sites for the myosin heads are now made unavailable to the myosin heads. Therefore, cross bridges cease being formed and the muscle relaxes. After you finish viewing the screencast, please watch the short animation synapse.mov, which illustrates how motor neurons stimulate skeletal muscles to contract. Let's now review the objectives of this screencast. Describe a motor unit. Explain how motor units differ in muscles with fine motor control versus in muscles with coarse motor control. Describe in detail a neuromuscular junction. Describe in detail how an action potential produced in a motor neuron causes contraction in a skeletal muscle fiber. And lastly, describe how a skeletal muscle fiber relaxes. The next screencast covers the topic of contraction of skeletal muscles.